This is DNA replication, one of the two processes that can occur after the cell goes through a replication
The first step of DNA replication involves helicase, which separates the double helix by breaking the hydrogen bonds, and the single-stranded binding proteins, which maintain the separation of the double helix.
As a side note, here we have two types of nucleotides, NTP and DNTP. Before a nucleotide bonds with another to form the backbone, it takes the form of NTP. The NTP then loses this oxygen to become DNTP. This oxygen opens up a spot on the 3' carbon to bond to more nucleotides. The same occurs with NMP and DNMP, except there's only one phosphate instead of the 3.
There are DNA and RNA.
Once the backbone is formed, the nitrogenous bases hydrogen bond, not covalent bond, to each other. The 4 nitrogenous bases are thymine, adenine, cytosine, and guanine. The thymine always bonds to the adenine, and the cytosine always bonds to the guanine. This is how the two backbones come together to form the double helix.
In the second step of DNA replication, new RNA nucleotides are added by RNA polymerase. New DNA is added by DNA polymerase, better known by its street name, primase. It is important to note that the new RNA nucleotides can only be added to the 3' end of the carbon. This new fragment is known as an Okazaki fragment. This whole process is considered dehydration synthesis.
In the third step, RNA is replaced completely by DNA polymerase. In the fourth step, ligase attaches broken DNA.
In contrast to the transfer of
Something that is happening constantly is DNA gyrase. This is when the enzyme topoisomerase cuts one strand of the DNA, unravels it, and reattaches it to release tension and prevent the DNA from breaking. The leading strand consists of the new already connected DNA, while the lagging strand is attaching more DNA, but only attaching when moving forward.